Labor is a multi-stage process that requires the coordinated activation of intrauterine components, including increased myometrial contractility, cervical ripening, and membrane and decidual activation. Indeed, the transition from the quiescent to a contractile myometrium heralds the onset of labor. Histology, targeted RT-PCR, and bulk transcriptomics have been applied to investigate tissue-wide myometrial changes associated with contractility. However, the uterus represents a heterogeneous cellular landscape, and thus, a more comprehensive investigation at single-cell resolution is required. Herein, we utilize single-cell RNA sequencing to establish a cellular atlas of the human myometrium at term parturition. Tissue samples were derived from the lower uterine segment of 13 laboring and 11 non-laboring women, single-cell suspensions were prepared, and single-cell RNA-seq was performed. 24 cell types were identified, highlighting the cellular heterogeneity of the myometrial tissues at term pregnancy. We evaluated the labor-associated changes in gene expression across all identified cell types. We found that multiple cell types displayed significant changes in gene expression with the onset of labor. Correlation analyses of labor-associated gene expression demonstrated strong associations among major cell types, namely endothelial, smooth muscle, stromal, and myeloid cells. Notably, we identified 3 distinct clusters of smooth muscle cells, namely smooth muscle cell 1, 2, and 3. Smooth muscle cell 1 participated in smooth muscle contraction, smooth muscle cell 2 was related to neutrophil function, and smooth muscle cell 3 was enriched in interferon gamma signaling, all of which were validated using immunohistochemistry. Therefore, single-cell RNA-seq identified smooth muscle cell subsets with distinct functions in labor. Next, we further leveraged our data to explore cell-to-cell communications during the process of labor. Both immune and non-immune cell types can serve as senders and/or receivers of labor-associated signaling pathways, which included interleukin-1 alpha, TNF, contraction, and complement. Therefore, these data indicate that both immune and non-immune cell types participate in the complex signaling of human parturition. Next, single-cell signatures derived from the myometrium were intersected with the transcriptome of the maternal circulation to show that both immune and non-immune single-cell signatures can be monitored non-invasively throughout pregnancy. Next, to enhance the relevance of monitoring myometrial signatures in the circulation, we evaluated labor-specific transcriptomic changes shared between the local and peripheral compartments. Notably, myometrium-derived signal-cell signatures, including monocytes and macrophages, were enriched in women who underwent spontaneous labor at term, providing a potential non-invasive means of monitoring this process. In summary, we utilized single-cell RNA-seq to establish a single-cell atlas of the human myometrium and to characterize the cell type-specific transcriptomic activity and cell-to-cell communications that are modulated during the physiologic process of labor. Notably, myometrium-derived single-cell signatures can be monitored in the maternal circulation throughout pregnancy and are enriched in women with spontaneous labor at term. This study was published in JCI Insight and was conducted by the Perinatology Research Branch, Division of Obstetrics and Maternal Fetal Medicine of the Intramural Program of NICHD, NIH, as well as the Perinatal Research Initiative of Wayne State University.